So I'm hoping this will work. Okay, so diversity of Archaea today. My hope is that I will be able to go over diversity of viruses because I kind of moved that lecture to this class because I completely reduced diversity of Archaea, so you guys don't have to worry too much. That's why that means we have enough time for other more interesting stuff. Anyone? So everyone ready for quiz? 1 hour. So you have 1 hour. I extended time comparing to syllabus. You have 1 hour, no lecture that day, sit and write. It won't be hard. So, and let's see how it will go. But I do have, and I was telling a few minutes ago, I do have to separate people who are A+ from people who are C-. That's pretty normal. There is nothing wrong with that. So, yeah, and there are people who are doing quiz 1 as well. So anyway, Archaea. 5 phyla. Yes, sorry, yes.
Yeah. Will you have bonus questions on
this? Yes. I always have bonus questions. And I have an idea because you have guest lecturer coming on November 2nd. So because he will make 1 question for the midterm. I'm thinking this is just idea. I have to ask Nora, so I still cannot confirm. This is unofficial. Basically, whoever comes on the class will have 50% of that question on midterm. So that's just my idea. I have to ask Nora because I want to I want to help somehow. I want to give some sort of award to people who come for guest lecture. So, but I have to confirm with TA. I don't know. So, Archaea 5 phyla. Euryarchaeota, Traumarchaeota, Nanoarchaeota, Krenarchaeota, and Corarchaeota. I don't care to see your writing. I want you to know how to recognize these phyla. So, are you going to pronounce them correctly? I probably don't pronounce them correctly. I just want you to know what main characteristics of these 5 phyla. And to be quite honest, Nanoarchaeota are quite interesting and they have only 1 organism. The same for Corarchaeota, only 1 organism, the 2 ones we know. Traumarchaeota, ammonia oxidizing archaea. So we talked about most of them. And now, in terms of phylogeny for Archaea, this is their phylogenetic tree, which Nora will show you. Nora will show you how to make this. This is the most basic phylogenetic tree, and it shows where each of these phyla is. So there are phyla, and they're separate branch, as we know, based on 16S rRNA. And apparently, evolution of Archaea is driven by horizontal gene transfer that happened between these phyla. They're very closely located on phylogenetic tree. And some of common traits that are shared in all Archaea, like regardless of is it Nanoarchaeota, Euryarchaeota, Traumarchaeota, Krenarchaeota, they have ether-linked lipids. And this is the basic feature that I will always ask you to know. And this is one of the main differences between Archaea and bacteria. Bacteria have ester lipids, Archaea have ether-linked lipids. Another thing is that Archaea do not really have peptidoglycan in cell wall. Archaea have all sorts of variations in cell wall. Like it's not peptidoglycan, they have pseudopeptidoglycan. Some of them have pseudopeptidoglycan or pseudomurane, but it's not real peptidoglycan that you see in bacteria. Another feature that makes Archaea quite interesting, and you will see at the end of the class, we now know that Archaea are actually highly closely related to eukaryotes, is that all molecular machinery, like replication, transcription, translation, so I have here example of RNA polymerase, is very much resembling eukaryotic structures. And if you remember first few lectures, we had that table that talks about bacteria, archaea, eukaryotes. You will see that archaea sort of have features from bacteria and from eukaryotes. So, and some of them are very unique for archaea, but replication, transcription, and translation are pretty much resembling eukaryotic structure. Which and nowadays they actually found even more evidences of eukaryotes and archaea connections. In terms of metabolic diversity, they're really diverse, and they usually have extremophilic metabolism, which means they're adapted to unusual environments such as high temperature. Chemoorganotrophic, chemolithotrophic, anaerobic, aerobic. So where I'm going with this is that they're highly diverse. Literally, almost everything exists in Archaea. It's not everything. Euryarchaeota, so the way how you will memorize this is like European Union, EU, Euryarchaeota. So when I studied, they taught us that these are actually real archaea, like Eury stands for real. That's not really true, but that's how I memorized it. And they can be salt-loving, halophilic, or they can be methanogens. So we already talked about methanogens as one of the most ancient metabolism, or they are hyperthermophilic, which means they live at high temperature. So these are not bacteria. Last class we talked about evolution of life at high temperature, but this is in archaea, not in bacteria. Or they can be acidophilic, which means they like low pH. Halophilic organisms are those that really live in high salt environment. And usually archaea are extremophiles. That means that they're really highly adapted into the extreme conditions. So there are some that are non-extremophilic, but these salt-loving are extremophilic. So two organisms, the way how you will always recognize archaea that likes salt is halo. So halo, that's pretty much halo focus, halo quadratum. So all what you need to know is that prefix halo. Halo tells you that it's salt-loving organism. Usually they are aerobic, so they are still extremophilic. They live in high salt, but they are aerobic, and they can use organic compounds, usually amino acids, but it will be enough to know it's organic compound. And they don't do fermentation, which is interesting. They just do anaerobic metabolism. They respire nitrite, so aerobic, anaerobic, but no fermentation. This is strictly for archaea, for this archaea. Some of characteristics is that Halococcus has very large plasmids, so pretty much largest in nature, high percentage of total cellular DNA, while the other one, Quadratum, has those gas vesicles. So if you remember, we talked about cyanobacterial gas vesicles, so these guys also have gas vesicles, but and the purpose is the same. So in cyanobacteria use gas vesicles to float, but that's how they search for the light. These guys have those gas vesicles that help them float in those highly salty environments. They do have very specific mode to balance intracellular solute concentration comparing to in that high salt environment. So if you think about that, high salt environment has a lot of solutes dissolved. So what organism has to do in that environment is to accumulate or synthesize some solutes inside of cell, so it can balance, because there are so many salts outside, and then if there is no enough salts inside the cell, all salts from outside will diffuse and then can poison cells. So what the organism is doing is making solutes inside so it can balance that outside concentration of so that's called compatible solutes. So compatible salts are literally preventing dehydration under osmotic stress, and high osmotic strength means a lot of salts in environment, like high salts. One of characteristic of Archaea is that they have this is very unique for them. So instead of accumulating organic salts inside of cell, they actually do import of potassium from outside. So potassium is here analog to the salts, and that import of potassium is balancing the level of salts outside. So that's very unique for Archaea. For Archaea, usually it's just compatible salts like trehalose or another or salt or sugar, anything that can balance that concentration. Another group of Archaea are high temperature loving, which you can know, which are known as thermoorganisms. Like I say, thermo, using literally prefix thermo. So again, I'm never going to ask you to distinguish Thermococcus thermoplasma. I can ask you to distinguish Thermococcus and ferrooxidans. So bacteria Acidobacillus ferrooxidans versus Thermococcus. So ferrooxidans does iron oxidation. This one is high hyperthermophilic. So that's all what you need to know. So I'm trying to facilitate the memorization of some names because I want you to understand. So thermo means hyperthermophilic. And thermoplasma is the first one that I will cover here. And just think about this thermoplasma. Plasma means there is no cell wall. Think about mycoplasmas, ureaplasmas. So those are bacteria that don't have cell wall. Actually, don't have cell wall. This is Archaea that does not have cell wall. And it has very small genome. That's pretty much, again, characteristic of mycoplasma and ureaplasma. Grows in literally, we say 55 degrees at very low pH, but this is high temperature. 55 is high, not so much as 120, but it's still high. And there are no sterols to stabilize it at high temperature. Usually sterols are there in those organisms that live at very, very high temperature, like 100 and something, but this one doesn't have it because 55 is not really high. One thing that I didn't mention is that it uses sulfate respiration, but for this one, it's really only important to know that is hyperthermophilic cell wall lacking organism. The next one is methanogens. So methanogens we already covered multiple times, and the name tells you methanegenesis, so something that makes methane. And they are strict anaerobes, which means they live with no oxygen. And we talked about that in the first portion of the course when we talked about the first metabolisms that developed in early anoxic Earth. So probably methanogenesis was even older than anoxygenic photosynthesis. So, and this methanogenesis is actually part of their metabolism. So they generate methane as a consequence of their metabolism. That's their product. And apparently we now know that actually those methanogenic archaea are actually one of the biggest sources of methane in the environment. They will be very present in rice paddies, but good thing is that since they live in an anaerobic environment, like on the bottom where there is no oxygen, they make a lot of methane, but then on the top, analog to Vinogradsky column, on the top, there will be methanotrophs that will literally consume that methane before it reaches atmosphere. So you have methanogens as a source and methanotrophs as a sink for methane. So if we are normal human population and we didn't really make so much industry, we wouldn't have problem because those two organisms, methanogens and methanotrophs, function in perfect collaboration. And Please don't ever try to pronounce this organism name, because I don't know how to pronounce it, and I'm not ever going to ask you that. What is important the only thing that you will have to memorize is methanol. So if something is methanol something, that's methanogen. Methanotrophs are quite different. They don't have their methylomicrobium, methylocystis. They're not methanol. When something has methane in practice, that means it makes methane. And the book says that this is model methanogen. I don't really think that this is model methanogen, because papers that I read when I was getting ready for grad school, I never found this organism. So this is just strictly from book. That's why I'm not asking you to memorize. But what is important, that what I want you to know is that there are all sorts of enzymes for methanogenesis in these methane-producing organisms, and the most important is that central carbon and metabolic pathways are very similar to bacteria, but transcription and translation genes are very similar to eukaryotes, pretty much like all Archaea. And last but not least is acidophilic organism. And as you know, we already mentioned this we mentioned ferroplasma when we talked about iron oxidizers. You remember when we talked about iron oxidizers, acidophilic, neutrophilic, and then phototrophic iron oxidizers. So this one is one of them. So ferroplasma, ferro stands for iron. And this picrophilus is something that we never mentioned before, but this one is quite unique. So picrophilus, which you will see now, belongs to Archaea that literally lives on pH 0.7. So lowest possible pH that exists in this universe. It is phylogenetic relative to ferroplasma. Iron is electron donor for both of them. That's when we talked about that iron oxidation, like ferro. But ferroplasma, and you see, plasma means cell wall lacking. So each of these names actually has a meaning. So ferroplasma is strong acidophile, but nothing is so acidophilic as picrophilus. It's 0.7. So that's like huge high low pH. I didn't know before this class, I didn't know that this organism exists. It's quite interesting that something actually can live in such an acidic environment. And hyperthermophilic organisms, which is again Thermococcus. I'm going a little back because there is this specific organism that is Methanopyrus. That is one and only methanogen that is hyperthermophilic. So, and again, methano. So that's how you can always recognize it. So methano means producing methanes, and pyrus, like fire, which means like high temperature. That's how you will know. And this methanogen is quite unique, and again, it has combination of bacteria and archaeal features. In Archaea, you will see that mixtures in between different domains. They're quite interesting because of that. So you will see that they have very unique. This Methanopyrus has very unique lipids, and they do have glycerol side chain, like Archaea, but they have phytanyl. So, but they also, because glycerol has like 3 hydroxyl group, it's 3 hydroxyl alcohol. So what you usually see is generally in bacteria you have added fatty acids on glycerol. In Archaea you have isoprenoids or something else. This one has mixture of fatty acids and this unique phytanyl. So it's like a mixture between bacteria and Archaea. And you will usually have these unusual lipids in the membrane that helps organism to survive at high temperature. And this phyla is very easy. Trauma Archaeota. I always call this Trauma Archaeota. I have a really hard time avoiding that R when I'm saying it, but that's why I'm not insisting on how you will say it. but this, how I call it, trauma archaeota, they give you trauma. Trauma archaeota are ammonia oxidizing archaea. And we talk about ammonia oxidizing bacteria a lot. Those are nitrifying bacteria. They oxidize ammonia or nitrite. Ammonia, so archaea from the phyla trauma archaeota, they also oxidize ammonia. What distinguishes them from bacteria is that they actually have ability to detect teeny tiny amounts of ammonia, which bacteria don't. And the way how they do it is because that enzyme, ammonia monooxygenase, the enzyme that we talk about that is highly evolutionary, similar to methane monooxygenase. So that enzyme in ammonia oxidizing archaea has much higher, 100 times more affinity for ammonia than bacterial ammonia monooxygenase, and they're way more efficient. And you will see them in oceans usually. They grow at very, very low concentration of ammonia. They are very unique because they have this CREN-Archaeol lipid. But that's not so important for this class. It is important to know the key difference between ammonia oxidizing archaea and bacteria, and that's in the affinity for ammonia. Archaea have enzyme that has much higher affinity for ammonia, and they are mostly present in oceans where they control nitrogen cycle. And this one is extremely cute, and the name is very cute. It's called Nanoarchaeum equitans. So does somebody know what equitans means? What do you think? Yes, but in terms of animals, horse? Equum, yeah. So it's a Latin name for equus. So I'm probably they're isolated from some horse or something. But what is really cute is I don't really know. I'm just guessing, but it's a very small genome, and that never grown in pure culture. So this Nanoarchaeum actually lives in symbiosis with this host organism Ignicoccus hospitalis. And what this means, Ignicoccus hospitalis, is actually a name that means hospitable fireball. So Ignicoccus hospitalis is another archaea from another phyla that literally hosts this nanobacterium. So nanoarchaeum, is literally attaching to the surface of Ignicoccus, and there are multiple cells of nanoarchaeum to Ignicoccus. So, and it's called hospitable fireball because it's hyperthermophilic. So, and there is only one organism in this phyla. So both of them, and Nanoarchaeum and Ignicoccus, live at high temperature, 90 degrees. So basically you have like multiple cells of Nanoarchaeum are on Ignicoccus. And why is this symbiosis? Well, Ignicoccus literally does, Ignicoccus, that CREN-Archaeota, that's a host, like hospitable fireball. It's doing hydrogen oxidation and respires sulfate, and does carbon dioxide fixation. But this Nanoarchaeum is completely dependent on Ignicoccus. So it cannot grow without it. And when they, of course, they sequenced the genome, and they noticed that genes that remain in Nanoarchaeum equitans are literally genes for lipid biosynthesis and for some molecular machineries like DNA repair and enzymes, and replication, transcription, translation. But they actually lost all genes for amino acid biosynthesis or nucleotides or coenzyme. They even lost ATP production genes. So I forgot to move it here. So interesting thing is that almost all genome of Nanoarchaeum equitans codes proteins. So I didn't really understand that. And I did not find in literature what kind of use is Nanoarchaeum for Ignicoccus. So I don't know. but that's something that maybe we can search because it's symbiosis. So it must be they do something to help each other. So Ignicoccus gives carbon dioxide fixation compound, but what nanoarchaeum gives, that I don't really know. So just question to think. Core Archaeota, again, there is only one organism, and it's called cryptic filament of youth. I don't know why it's called like that, but what you need to know is that it's actually hyperthermophilic, and it does not have ability to synthesize molecules for its own growth. So it lost certain genes. And this organism, this is the most important to know, that this organism from Core Archaeota is the example of black queen hypothesis. Does somebody know what is black queen hypothesis? Did you hear about that? It's interesting way how they described evolution and why genes are lost. So this member of Core Archaeota cannot synthesize some of its main molecules for the growth, and this is called evolution of mutual dependence, or black queen hypothesis. So basically, this hypothesis describes the strategy, evolution strategies, where one organism loses costly functions, like loses the genes that spend a lot of energy, but in the favor of gaining genes that improve growth efficiency. So basically, the result is, and this is directly from a book, result is a mutant cheater that becomes obligatory dependent upon provider. So provider would be black queen. So somebody who has to give the required molecules that this organism lost so they can function. So black queen is the provider. This was a fun fact from the book. And Cran Archaeota, we already talked about Ignicoccus hospitalis. They are autotrophs and primary producers in extreme photosynthesis, in extreme ecosystems. So there is no so the only specific organism is Ignicoccus, which we talked about when we talked about nanoarchaeum equitans. Any questions before we move to slightly harder portion? There are only a few. So, okay. And so next few slides, I just wanted to use to describe you the importance of connection between Archaea and eukaryotes. And there are two models that were describing that connection. And we already kind of mentioned this connection when we talked about two hypotheses of how first cell developed. So one of them was, the first model was oocyte tree. So what means oocyte? Oocyte means something that has no nucleus. It doesn't really matter who was, I'm not going to ever ask the name of the scientist who developed this hypothesis, but you should know how to recognize this unrooted tree. And this will be important for midterm exam, just to recognize. I'm not really asking you to draw. This is actually abandoned. Nobody makes this kind of phylogenetic trees. When Nora shows you how to make phylogenetic trees, she will show you rooted tree, like, so you can see where is the common ancestor, where everything starts branching. So based on this hypothesis, the ancestor of eukaryotes was the organism that could thrive at high temperature, but did not have nucleus. So basically the ancestor of eukaryote was oocyte, which is like here. Then you have a branch one, which is here, when eukaryotes develop, and those are karyocytes. So karyo means nucleus. So that's branch one. The branch two from oocyte is basically branch that gives bacteria and archaea and goes here, and then they branch to L bacteria, which are real bacteria, and Archaebacteria, which are old ancient bacteria, like archaic. So that was the first. So you can already see that this doesn't really make sense. Why? Because you will see later when we mention those specific archaea that were recently discovered. Archaea are definitely way more connected to eukaryotes than to bacteria, and that's why this 3D tree developed. This is the second model, where literally the hypothesis was that you had prokaryotes that were branched at two domains, bacteria and archaea, and then eukaryotes separated from archaeal branch as a separate lineage. And all biochemical data and data that we have from DNA and RNA and the available knowledge in molecular machinery, so all three domains, really tells us that bacteria are not so similar to archaea how eukaryotes are similar, especially in terms of molecular machinery. So all data, biochemical data, go into the favor of this hypothesis. So we now know that eukarya and archaea have common ancestral branch, and the branch that gave rise to eukarya and archaeotes is different than the one that gave rise to bacteria. Even though bacteria and archaea are both prokaryotes, it's quite surprising how much eukaryotes and archaea are similar, while archaea are actually distant relatives of bacteria. So this is important. So basically, we say that archaea and bacteria are not monophyletic group. So what means monophyletic group? So did you guys hear about that? Did you have any evolution classes? Yeah, so you know, so this was not initial in my lecture, but I realized that there might be people who never heard about monophyletic, polyphyletic, and paraphyletic groups. So I cannot really tell you that bacteria and archaea. Yes.
So are, like for this slide, you're saying bacteria and archaea are sister groups with bacteria as the outgroup?
Bacteria and archaea are not, how do you mean outgroup?
Like eukarya, archaea, and then bacteria up here. Separate.
Not exactly. Common ancestor, bacteria, archaea, eukaryotes.
Okay.
Okay, I am guessing that if I have like a board, like if I can, so basically this is where your common ancestor is, okay? And then you have bacteria here and archaea go here, okay? And then from there you have eukaryotes that are distant. So literally, if you compare, archaea and bacteria are distantly related, but bacteria and but archaea and eukaryotes are actually way more closely related than bacteria and archaea. That's why we say that they're not monophyletic group. And I'm not going to ask this in exam because that's a really, you probably heard about this in evolution. Maybe Nora will talk about that in her lectures. But I just wanted to remind people, just to explain what means monophyletic, what means paraphyletic, what means polyphyletic groups. So again, bacteria are here, not monophyletic groups. So what is monophyletic group? That goes in the blue. So the groups of organisms that are closely related to each other, and this is where they have common ancestors. So they all, three of them, all from the same ancestor. When something is paraphyletic, this is the green one. So this is the group that includes single common ancestor at some point of groups. So some of them, so these are paraphyletic. So they don't, so this and this don't have common ancestors. So this is ancestor of this is ancestor of that. But at some point they had common ancestor. And polyphyletic means no recent common ancestors. So just a brief reminder so you can understand what means monophyletic, polyphyletic, paraphyletic, but this is not going to be an exam. This was just me reminding people who maybe don't have that knowledge. But not going to be on our exams. And again, main point, very, very close. Archaea and eukaryotes. So, and this actually happened in October last year when those Did you guys hear about Asgard? This should be Asgard group of Archaea. Actually, Asgard. So Asgard group of Archaea are a novel discovered Archaea film, and I have actually very good study on this. It was just from last year. So what they found in these Archaea is a huge amount of evidences about extremely close relationship between archaea and bacteria. So in Asgard archaea, they found this endosomal sorting protein complex that exists in our organism. So that's they found homologs. So that was so unique that never existed in any other prokaryotes. And there are homologs in these Asgard archaea for cell trafficking protein. So cell trafficking is pretty much common in eukaryotes because they have such a complex transport signaling. And Asgard archaea have homologs of these proteins. This was a huge discovery, and they belong to. So it doesn't really. I'm not going to insist in which phyla they belong because I do think that eventually they will reclassify Asgard archaea as a separate phyla, not as Archaeota, because of they're so unique and so specific. So this ESCRT machinery that was found in Asgard archaea those are all homologs of eukaryotic complex signaling mechanism. So this just shows that archaea and eukaryotes are very closely related. And again, you don't really have to know details from the paper. If somebody wants to read, I'm going to give you that paper for sure. Before we move to summary, does somebody have questions? This will be just a brief summary, and then we have a short. Yeah, you have questions. Sorry. No. Okay. Sorry. So, brief summary. Again, I'm not going to go in details. From this lecture, I do want you to know only a few important things that will be in review lecture that I will post before midterm, and that are halophilic archaea need a lot of salt to grow. So salt, sodium chloride, that's all what you need to know. They live in, actually not they live in an area where there is a lot of salt, and the way how they cope with high salt is by importing potassium in the form of potassium chloride. Methanogenic archaea have metabolism that is linked to the production of methane. Usually it's an ancient metabolism. We talked about that, hydrogen plus carbon dioxide. Extremely acidophilic picrophilus ferroplasma. I don't think I will ever ask thermoplasma as a part of acidophilic response because that one is also hyperthermophilic. Ferroplasma does lack cell wall, and they remind on mycoplasmas in that term. The one that I really like is Methanopyrus. Apparently, this one is a recorder, hyperthermophilic methanogen, apparently grows at 120. So this is the highest ever known temperature. Taumarchaeota are ammonia oxidizing archaea that have ammonia monooxygenase with extremely high affinity for ammonium, and they are primary producers in oceans. Nanoarchaeum equitans is the only organism hyperthermophile in Nanoarchaeota film that lives together with Trenarchaeota Ignicoccus hospitalis, and it has teeny tiny genome because it lives in symbiosis. Cryptic filament of youth, Corarchaeota film, it's hyperthermophile. That is an example of black queen hypothesis. So then I have a few reminders about the life evolution of life at high temperature, and that is above 150 degrees, there is no life because key molecules like ATP are destroyed after 150 degrees. The recorder is 120 degrees. Again, a few key points about the life on, we talked about that in last unit, that life at high temperatures. Basically, they have all those adaptations, specific folding, specific protein folding. So it's not about specific amino acids, it's about specific protein folding that help them to survive high temperature, specific DNA binding proteins, specific lipids that, so basically molecular adaptations that help them to survive at high temperature, and molecular adaptations refer also to DNA and RNA. So that's another thing. And this is pretty much what we already talked about. Hydrogen metabolism as one of the earliest forms of metabolism evolved on Earth. In the last class, we talked about that specific hydrogen oxidation enzyme HAC that has very specific adaptation, specific gas channels that enable them to exclude oxygen and detect literally teeny tiny amounts of hydrogen, so they can even oxidize amounts of hydrogen that are present in the atmosphere, but that's not going to be on quiz. And that's all for Archaea. My next lecture is about viruses, but